AS Level Biology 9700, Paper 1, Variant 1, May June 2025. And today we are going to discuss from question number 1 to question number 13. And question number 1 is, which statements about light microscopes are correct? They have given us 3 statements and we have to find out which one is correct, or all these statements are correct. So calculate the magnification of a light microscope. The eyepiece, lens and objective lenses magnifications are added together. Not, this is not a correct statement because they are not added together. So the magnifications of the eyepiece lens and objective lens are multiplied. So they multiply together. So this is not the correct statement. The second is, the resolution of a light microscope is limited by the wavelength of light. This is correct statement because the resolution of the light microscope is, you can say, is changed as the wavelength of the light is changed. So I would like to say that the light of shorter wavelength uh, of the light microscope will show the better resolution. So this is the correct option. The divisions on the scale on a stage micrometer are closer together than the divisions on the scale on an eyepiece reticule. So here you must know that the stage micrometer is that scale which is kept on the stage in the form of a slide, and whereas the eyepiece reticule scale is always kept in the eyepiece. So the divisions on the stage micrometer are closer together than the divisions on the scale on an eyepiece reticule. So they have, so this is not a correct option. They are not closer. They are far from each other. So stage micrometer has a fixed scale and the scale of the eyepiece reticule is changed as the magnification is changed. So this is not a correct statement. So only statement number 2 is correct, and which is answer is C. A, B and D are not correct options. Only according to these statements, only the C answer is correct. Now we will move towards the question number 2. And here question number 2 is, a student measured the width of a mitochondrion in an electron micrograph of an animal cell with a magnification of 9600. The width was 6 millimeter. So first of all, I'm going to write the formula for this as magnification is equal to width of micrograph over actual width. So they have given us magnification which is 9600. So we have to find out the actual width. So actual width will equal to width of micrograph over magnification. But the width which they have given us is 6 millimeter. They have measured it with in millimeters units. But to find out the actual width, we have to change this 6 millimeter into micrometer. So we will multiply 6 with 1000. So it will become equal to 6000 micrometer. So we have to find out the answer in micrometer, because in all options they have, and in AS levels, we always measure the magnification in micrometer. So Actually, there is no unit for magnification. Actually, you want to say that if we are measuring the width or length or diameter, so it should be in micrometer unit. So actual width will be equal to it is 6,000 by 9,600. So it will be equal to 0.625 micrometer. And according to this, the option B is the correct option. So you have to find out this with the help of this formula. Now I'm moving towards the question number 3. The question number 3 is the photomicrograph shows a section of the stem of a plant. Which statements could describe you? And you can see this tube is the xylem cell. So the cell wall contains pits. This is the characteristic of the xylem wall, that they have pits because the lignin is deposited in the form of pits or in any other form. There are two more forms for the deposition of this lignin, but they are talking about the pits. So they are present in the xylem. So the cell wall contains cellulose and also lignin. Cellulose and yes, cellulose is also present and lignin is also deposited in the wall. So according to this, you can see the pits are thin areas in the cell wall that allow water to move laterally between adjacent xylem cells. So these xylem cell walls contains this cellulose as well as the lignin. So I would like to say the cell walls of xylem vessels are strengthened and they become waterproof with the help of this lignin. So in addition to the standard plant cell wall component, cellulose, lignin provides structural support. So lignin provides structural support. So this statement is also correct because the lignin is also present there. It is connected to adjacent cells via plasmodesmata. So no, this statement is not correct. The plasmodesmata, with the help of plasmodesmata, two cells are joined with one another. But in this case, in the case of xylem cells, the two cells, xylem cells, are not joined together with the help of plasmodesmata because they lack a protoplast. They don't have cytoplasm and nucleoplasm, due to which they are not joined with the other cells with the help of plasmodesmata because the lignin is a dead material. So that is the reason the statement number 3 is not correct statement. According to this, statement number 1 and 2, both are the correct statements. So according to these statements, the answer is A. B, C, and D are not the correct options. Now I'm moving towards the question number 4. And question number 4 is the diagram is taken from an electron micrograph of a cell that secrete enzymes. So where are the polypeptides for these enzymes made? Polypeptides, the protein chains which makes the enzymes. So you can see the 4 different options has been given over here. So according to this, according to these, you can see, I would like to say C is the correct option because this is endoplasmic reticulum. On endoplasmic reticulum, the ribosomes are attached. So they are rough endoplasmic reticulum have these ribosomes on them. So that is the reason I would like to say A is the secretion of and B is the Golgi apparatus. And you can see that D is the mitochondria. So ribosomes are helpful for the, you can say, formation of enzymes or protein synthesis takes place with the help of these ribosomes, you know. So C is the correct option because the ribosomes are present on rough endoplasmic reticulum. Question number 5 is, which comparisons between a typical bacterial cell and a typical plant cells are correct? So the first option, first statement is cytoplasmic DNA and nuclear DNA. Yes, this is correct. The bacterial cell don't have a proper nucleus. So the DNA is present in the cytoplasm in the form of circular rings. Second, nuclear DNA in plants, the proper nucleus is present. That is the reason the DNA is present in the nucleus. Then no smooth endoplasmic reticulum present. Yes, uh, this is also correct. Smooth endoplasmic reticulum are present in plant cells. Both types of endoplasmic reticulum are present, smooth endoplasmic plus rough endoplasmic reticulum. So in bacterial cell, smooth endoplasmic reticulum are not present. Peptidoglycan cell wall found. The cell wall of the bacteria is made up of peptidoglycan instead of cellulose. So this is correct. Cellulose cell walls are present, that is the cellulose cell walls are made up of in the plant cells. So they are present in these cell walls. Cellulose is present in the cell walls of the plants. The fourth, 80S ribosomes and 70S ribosomes. There are 70S ribosomes, but in plant cells there are 80S ribosomes. So this is not the correct option. Number 4. So according to 1, 2, and 3, they are the correct statements. So according to them, A is the correct option. Now I am moving towards the question number 6. And question number 6 is, the diagram shows a virus. And here you can see uh, they have labeled the 4 parts. And according to them, they are W is DNA. You can see a double strand structure is there. So this is DNA. And X is phospholipids. Whereas Y, the nuclear capsid, is made up of proteins. And Z is also a protein. So according to this labeling, we can see This is not RNA. RNA. So A and D are totally incorrect options. B or C can be correct, but because there is DNA. But according to this labeling, which I have done, you can see the B phospholipids, they are there. They are made up of phospholipids. So proteins, Y is protein and Z is protein. According to this, B is the correct option. So X is not the B is the correct option. A, C, and D are not the correct options. Next, question number 7 is the iodine test and the Benedict test were carried out on samples of a starch solution. Starch solution, and you know the test for starch is iodine is used for this purpose. Amylase was added to another sample of the starch solution and incubated for 10 minutes. The Benedict test was then carried out on this sample. To break down the starch, the amylase enzyme is used for, they have used amylase over here because the amylase is that enzyme which digests the, which break down the carbohydrates. So what were the results of the test? Iodine test before adding amylase. So it will be positive because the iodine test is used for starch. So A and D are not correct options. So it can be B or C. So it can be B or C. This is correct. So Benedict test before adding amylase. Benedict test is used to find out the presence of reducing sugar. So Benedict test is for reducing sugar. And iodine test for starch. So reducing sugar. Starch before adding amylase. It means now the starch is there. Then starch will be metabolized and it will change into reducing sugar. You know, they are monosaccharides. Glucose, fructose. So in this way, this is not the correct. Benedict test after adding amylase, it should be positive. So this is, so according to this answer, that is when, let me explain again. When we add the iodine into starch solution, then it will change into blue black, which will show the starch is present. Then we will add amylase into it and we will perform the test. So it will show that the starch is broken down into monosaccharides, and then it will, the Benedict, we will perform Benedict test and Benedict test will give positive results. But when before adding the amylase solution, if we perform the Benedict test, so it will give the negative results. So according to this, C is the correct option. C is the correct option. A, B, and D are not the correct options. Now we will move towards the question number 9. Which row about alpha glucose and beta glucose molecule is correct? So, you know, alpha So this is alpha glucose and this is beta glucose. And you can see OH present in the on carbon number 1. So this is the carbon number 1, 2, 3, 4, 5. So the carbon number will be 1. So according to this, C and D cannot be the correct options. It is on carbon number 1. Cellulose contains both molecules. So which row about alpha group beta group is there? Atom on which the OH position is different. That is carbon number 1. And you know, this cellulose is made up of beta glucose only, not alpha glucose. Beta glucose is there in the formation of cellulose, which is a polysaccharide. So I would like to say A is not a correct option. According to this, the answer is cellulose contain? No, it do not contain both. It only made up of this beta glucose. So according to this, both molecules are not. Only A is the correct option. Let me actually only beta glucose is used in the formation of cellulose. Now the next is which molecules, so according to this, A is the correct option. Which molecules are monosaccharides? Monosaccharides. Ribose sugar, deoxyribose sugar, both we say pentose sugar. So they are made up of 5 carbon. And glucose made up of 6 carbon. Sucrose, so 1, 2, and 3, they are monosaccharides. Whereas sucrose is made up of glucose, and glucose. So it is disaccharide. So according to this, A is the correct option. A is the correct option. Question number 10. Many animals use triglycerides for long-term energy storage. They have given us 3 statements. Which statements are correct? Which statements are correct? No, number 1 statement is they provide buoyancy in small marine animals. This is correct statement because the triglycerides, they are the fats. They are less denser than water. So they are less denser than water, due to which it helps buoyancy, or they help the marine animals. They are the main components of cell membranes. Cell membranes don't have triglycerides. Instead of, there are phospholipids. Main components are phospholipids, in which the head of the phospholipids is hydrophilic, water loving, hydrophilic head, and hydrophobic tail. And both head and tail, they will make a lipid bilayer, not the triglyceride. And you know, triglycerides are made up of, these triglycerides are made up of glycerol plus 3 fatty acids. So according to this statement is not correct. They are used as thermal insulation. Yes, triglycerides are excellent thermal insulators. They provide the insulation, and they are present beneath the skin of mammals. So we human also have this layer of triglycerides. So according to this, statement number 1 and statement number 3 are the correct statements, and statement number 2 is not the correct statement. So according to this, C is the correct option. Now we will move towards the next question, which is the diagram shows 3 different bonds. You can see 3 different bonds are there. Which bonds are present in proteins? Here you can see a hydrogen bond. Hydrogen bond is a weak bond because it has been shown with a dotted line. So it is present in, and second bond is the peptide bond. Peptide bond, and peptide bond is always formed. It is, you can say, type of covalent bond. It present in the amino acids are joined together. On one side, carbonyl group, and the other, this is the carbonyl group, and other is the amino group. So it is also present. Third type is disulfide bond. So the disulfide bond is also present in protein. So this is also type of covalent bond. So this bond is also a covalent bond and is formed between the sulfur atoms of 2 cysteine amino acid residues. So it play a crucial role in forming and stabilizing the tertiary and quaternary structures of the proteins. So these 3 are present. So according to this, A is the correct option. Now we will move towards the question number 12. And question number 12 is, which statement describes the quaternary structure of a collagen molecule? A polypeptide that form an alpha helix which are held together by covalent bonds. Collagen molecule quaternary structure is not made up of a single chain. So alpha helix poly, this is a single chain in which covalent bonds are joined together. So I would like to say that collagen is a fibrous protein. So it has a high tensile strength. So its unique structure is key to its function in providing support to the tissues. So the quaternary structure of the is not made up of this. So the polypeptides that form a double helix again this is not the uh, two four polypeptides that are held together by covalent bonds which form a helix again. This is not a correct option because the only the three polypeptides that form helix structures which are held together by hydrogen and covalent bonds. So according to this, I would like to say that only D is the correct option. The three polypeptide chains, when held together with the help of covalent and hydrogen bonds, so they will make the overall stable structure. So only covalent bonds, and they are four polypeptides. So this is not correct option. So only hydrogen, they have shown only hydrogen covalent bond. Actually, there are three polypeptide chains. When they will join together, they will make, and they are with the help of hydrogen and covalent bonds. Both types of bonds are present between them. They will make a collagen molecule. So this is the correct statement D. Next, question number 13 is, the diagram shows the structure of the enzyme amylase, which level of protein structure results in the folding of amylase into its globular shape. So you can see the primary structure, is a linear sequence of amino acids. As for example, the amino acids, they will join together in the form of this. And in secondary structure, so they will join in this way. There will can be the bonding between them, and then in so they will little bit they will folding will take place. So when there are tertiary structure, it gives the 3D structure. Tertiary structure will give the 3D structure. So A, this is not B, and this is not quaternary. Because in quaternary structure, there should be the multiple chains. Multiple chains and multiple polypeptide chains. So to form larger functional protein complex. So D. So there is only tertiary structure which gives the 3D shape. So according to this in tertiary structure, the globular shape is formed, which can also be the 3D structure. So according to this, C is the correct option. It was all about from question number 1 to 13. Please like, share, and subscribe my channel and wait for my second video for this paper. Thanks for watching.